Okay, let's go ahead and get started. Good morning, everybody, and welcome to Grand Rounds. Our topic for today will come from the endocrine division. Before I ask Dr. Winters to take the podium, I just want to remind everybody that our departmental fall outing is occurring on Saturday the 20th at Huber's. It starts at 11. The food comes out at around noon. For those of you who've been to Huber's, you know what it's like. It's a lot of fun. There's stuff, if you have kids, there's stuff for little kids to do. There's wine. There's all kinds of little stores where you can buy all kinds of really absolutely great food. So we welcome anybody who wants to come. If you decide you want to come and you haven't RSVP'd yet, just let Jason know, just so we can get, some ballpark head count on how much food that we need. I don't have any other announcements. So with that, then, I will ask Dr. Winters to come up to the stage and introduce our guest speaker for today.
Thanks, Eleanor, and good morning. And it's certainly a pleasure to have Dr. Terry Davies visiting with us yesterday and today as a visiting professor. He's a professor and has an endowed chair at the Mount Sinai School of Medicine, where he was the division director for many years. Presently, he is the director of the program at the Beth Israel Medical Center downtown. Dr. Davies is initially from the UK, and after training in the UK in endocrinology, he came to do research on the G protein coupled receptors, the LH receptor, at the NIH, and that's where he and I met some 40 years ago. And after finishing his research stint at the NIH, he moved to Mount Sinai, where he has stayed for his entire career. He had always been interested in the thyroid, and of course the TSH receptor is another G protein receptor, so it was quite easy to take the biochemical methodologies that he learned as a laboratorian at the NIH to his new career, and he's been enormously successful. He's has a distinguished career with over 400 publications in the areas of autoimmune thyroid disease, understanding the TSH receptor, and antibodies to the TSH receptor, that is a Graves' disease. Dr. Davies has been the editor-in-chief of the journal Thyroid. He has written the chapter in the Williams textbook of endocrinology, which is the Bible for endocrinologists, about hyperthyroidism, a very comprehensive work. And he's been the president of the American Thyroid Association. So he's really very well traveled and gives lots of talks all over the world. And it's a pleasure to have him visit with us here in Louisville. Thanks for coming.
Well, thank you very much for having me here this morning. I've enjoyed myself so far. Unless any difficult questions come up at the end of my talk, I cannot for the life of me get this to switch on. So maybe it needs a new battery. So this is not a clinical talk. Thank you. I'm just warning you that we're going to take a quick move through autoimmune thyroid disease and look at some of the factors that we think precipitate the diseases. So this has broad implications, not just for autoimmune thyroid, but for any autoimmune disease. I'd like to think that the risks are very similar across the board. So if you have an interest in another autoimmune disease, a lot of what I'm going to say is going to apply. So I have no significant conflicts. So looking at autoimmune thyroid is very easy. It goes all the way from Hashimoto's thyroiditis all the way through to Graves' disease, and pretty much these two diseases are very similar. They just differ by the immune response to the precipitation, right? So that if you develop T cells or allow T cells to expand that are directed at the thyroid epithelial cell, you'll end up with apoptosis of the thyroid cells and Hashimoto's disease. If, however, the immune response is not so much T cell killing, but antibody mediated, then you'll end up with antibodies to the TSH receptor, which are thyroid stimulating, and this is Robert Graves' disease. And within the family, you can have both types of patients. Within the same patient, you can have both types of diseases swinging from one to the other. So they're very closely aligned. And one of the things we don't understand so well is quite how that choice is made. And that's what we're going to talk about. So the overall picture of autoimmunity is also pretty simple, right? There is a battle going on within the immune system between T regulatory cells, which control immune reactivity, and Th17 cells that secrete interleukin 17 that are pro-inflammatory and pro-autoimmunity. And the balance between those two certainly is important in controlling the degree of the immune response. And as I just mentioned, that response may take the form of either antibodies leading to Graves' disease or more likely T cell killing leading to Hashimoto's. So what precipitators do we know that are likely to lead us to develop one of these diseases? Well, we know a little bit about the genetics. Despite all the noise and all the money that's gone into genetic susceptibility in complex genetic diseases, right, those diseases where multiple genes are involved, we're actually still pretty naive, right? And even though we've recognized some, as I'll show you, some genetics associations, they are weak, incredibly weak. Environmental insults, or these days we don't say that anymore, we call it epigenetics. Influences from the outside are known. We're not going to have time to go through too much, but infection, radiation, smoke, iodine, trauma, the microbiome should be added on there also as important external influences on the immune system, some of which are transient, some of which can even be inherited. Thyroid antibodies we're going to try and deal with because they are clinically relevant. And pregnancy, I gave a talk yesterday afternoon on the role of autoimmune thyroid disease and pregnancy and how they're intertwined. And clearly they're very important. And we'll be able to review a little bit of that this morning. So let's start with something on genes. Now, I am hopeful that you'll leave here having learned something, even if you don't like the thyroid gland. So I'm going to try and stress a few principles as we go along that I think are worth staying awake for. Right? So as far as Graves and Hashimoto's are concerned, these are familial diseases. As I mentioned, they are complex genetic diseases in the sense that while you may have someone in the family with it, you can't predict who's going to get it. It's not Mendelian, right? So each gene is a Mendelian gene, but the complex genetic diseases are unpredictable. In clinic, about half your patients will give a history, a family history, if you ask. And it's an important thing to note down because it can help you with your diagnosis. What's the evidence in favor other than the familial clustering? Well, there is something called a sibling risk ratio. That is, what risk, what chance does your brother or sister have of getting the disease if you have it, right? So if the genetic risk is small, then your brother or sister's risk is only slightly increased, right? If, however, the genetic risk is high, then your brother or sister has a very, very increased risk of getting the disease. And that's called the sibling risk ratio. In autoimmune thyroid disease, it's quite high. 17 is quite high. And that suggests that there is a significant genetic component to the disease. And then, of course, there's the twin studies. I'm sure you know that twins, although they have a similar genome, they're not immunologically identical. All right. Do we have any residents here this morning? They're all busy doing other things. Well, it's a good resident question as to how twins differ in their immune response. No one's admitting to being a resident, right? So you've got to remember that the genes for the T cell receptor and for the antibodies, the B cell, the V genes, these genes undergo recombination. And it's the only genes that undergo recombination in the human genome, unless you have a cancer. Cancer cells can undergo that. But that means that as time goes on, the random recombination within the immune system means that twins, actually, their immune system becomes more and more different as they go through different experiences. Right? So the concordance between twins will never be 100% for any immune disease because their repertoires are different. Mm-hmm. However, in Graves' disease, it's about 30 or 40%, which is considered very significant, right, compared to non-identical twins, which is just a few percent. So there's clearly an important genetic component to these diseases, but we just don't know much about it. So how do you set a study to look at the genetics of complex diseases? So my feeling is that the literature and the New England Journal and those journals that you read have gone way ahead of what people really understand. So this slide is a sort of attempt to try and catch up a little bit, and that is to point out the difference between association and linkage. Right? I think it's very important to be able to understand the difference. So an association study is something that's relatively easy to do. So you would take a group of 100 patients with Graves' disease, and you would find 100 matched controls, and you would measure something in both groups. So if, for example, you wanted to measure the association of HLA, right, with Graves' disease, you'd measure HLA, DR3, say, in your 100 patients with Graves' disease and your 100 controls. And you would look at the difference, and you'd find that there's more in the Graves' patients than the controls. And then you would say, is this statistically significant? And if it is associated. HLA-DR3 is associated with Graves' disease. Now that is a very sensitive genetic test. And the statisticians say that you will detect any gene contributing a very small amount of genetic susceptibility. In fact, they think less than 5%. Right? Linkage is totally different. Right? Linkage is when you look at a family and say, well, who's got the disease and who has DR3? And you look at 2 or 3 generations or a lot of SIBs, and you see how often within the family DR3 is associated with the disease. And if it is significantly associated within the family, we call that a link, that it's linked. Now, that's a much more difficult study to perform, right? You've got to collect a lot of families and you've got to type everybody in the families. And so it's an insensitive system, right? And the genetic contribution, if you find something linked to a disease, it's very significant, right? It's really important, right? We don't have any linkages in autoimmune thyroid disease, let me tell you straight off, right? And you don't even have it in many of the autoimmune diseases. What you have is a lot of associations, right? And this can be by a simple test, as I mentioned, just measuring HLA-DR3, or it can be a whole genome study, right? A GWAS, right? A whole slew of SNPs, small little repeats within the genome that could be associated with your disease. And so you can do 1 million, 5 million SNPs, do a whole genome screen, and find what is associated. And that's what you read about all the time. And those associations, by what I've just said, are very weak. They're less than 5%, and the risk ratios are very small for them. So here's a whole genome screen on Graves' disease patients, 600 of them that I haven't published yet. These are 600 patients with eye disease, right? Remember that Graves' disease can affect the skin and the eyes because the antigen is widespread. And the only thing that comes up positive in a whole genome screen in Graves' disease is HLA, which we've known for 40 years, maybe longer, right? We didn't need all this new technology to find that. So as far as genetics are concerned, there are a few weak associations that I'll mention, many of which you can see here as sort of small dots in the distance, but there's nothing really powerful like HLA. And these are some of the genes that are talked about. They tend to fall into two types, one group is common and found amongst all autoimmune diseases. I think those are in red, right? And only two so far that are thyroid specific. The thyroid specific genes are thyroglobulin, which has a weak association with both Hashimoto's and Graves' disease, and the TSH receptor, which has a weak association with Graves' disease. But these are not impressive data, right? The other ones of interest, obviously HLA, that remains the most powerful association in almost all the autoimmune diseases. CTLA-4 up on the left, you may be familiar with. Antibodies to CTLA-4 are used in cancer these days to try and activate the immune response because CTLA-4 suppresses the immune response. Right? And CD40 is a co-activator of the T cell receptor. But all these associations are small. Now, this has nothing to do with autoimmune thyroid disease, but I do like dogs. So I have dogs. And so this paper from Science, looks like I don't have the reference on it, makes an important point about clinical phenotype, right? So in autoimmune thyroid disease, this is particularly important because patients turn up in all different ways, right? From hypo to hyper, for eye disease, no eye disease, right? Skin disease, pre-tuberous myxedema, all different. And this little paper, it's not so little, but this interesting paper made the point that 3 genes, right? FGF5 here at the top, RSPO2, KRT71. We don't need to know what they are really, but they are contributing to hair type, right? These 3 genes, by having variations within them, right, within the dog population, are responsible for at least 3, 5, 7 different types of hair, right? So different combinations of a small number of genes can make tremendous differences to the clinical phenotype, to how a patient will present. And I think we're only just beginning to get to the bottom of this type of interaction. So gene interaction, very, very important. Just a few genes differing can make huge differences to, in this case, curly hair or straight hair or frizzy hair. Now, the way HLA has become so critical is because it's not there for tissue typing. It's there for physiologic reasons, right? The HLA molecules are there to combine with peptides, antigenic peptides, that are then able to be seen by the T cells, right? So the T cell, which is on the right, has to be activated by seeing an antigen. And it only is able to see the antigen when it is complexed, right, with the HLA molecule. So this peptide, for example, might be a little piece of thyroglobulin, little piece of the TSH receptor, or if you're interested in type 1 diabetes, it may be a little bit of a beta cell, a little GAD peptide. And if you inherit T cells that are able to recognize these peptides, and that these T cells have escaped, right, from the thymus instead of being deleted, then they're going to be in the circulation, and when they see their antigen, they're going to become active, assuming there are appropriate co-stimulators that we don't need to go in too much to right now. There has to be co-stimulators to allow the T cell to become activated. Now, this slide is from a colleague called Jeroen Thoma, and he makes the point that particular forms of HLA, particular forms of HLA-DR3, like to bind particular peptides. So you have to have a DR, not just a DR3, but one with the right sequence to allow, for example, a TSH receptor peptide to bind. Right? And he just made the point here that at position 74, when there's an arginine, there is this particular negativity, and when there's a glutamine at 74, there's a different degree of charge. And so different peptides are able to go into this groove, right, depending on whether you inherit a glutamine at 74 or an arginine at 74. And I think it's a beautiful demonstration of how sophisticated the HLA system is and what you need to inherit. And this just makes that point that the risk ratio, right, for in this case Graves' disease, was much higher for the ARG74 versus the negative ARG74. Okay, so associations, but not linkage, have been shown in many populations, right, for HLA and autoimmune thyroid disease and many other diseases. But HLA genes have less than 5% contribution to genetic susceptibility. DR3 subtyping can sometimes improve that association, and here's an example at position 74. What about the TSH receptor, which is the primary antigen of Graves' disease and also involved in Hashimoto's in many of the patients? So it's a unique, when I say unique, it's a 7-transmembrane protein like many such receptors, but it's unique in having a very large ectodomain, the bit that floats outside the cell membrane. So let's take a look. I hope I have a picture of that. Yeah. Yes. Now, I don't know how to activate this movie. That's one of the, oh, there we go. So this is a little movie to keep you awake. Now this is the TSH receptor. See, I think this is beautiful. So here's the receptor on the right. This is the binding site of the receptor, and this is the antibody. This big thing, right? Antibodies are huge. Binding to the ectodomain of the TSH receptor. And you'll see it's binding almost exclusively into this concave area that is called the leucine-rich repeat area. And all the TSH receptor antibodies bind into this same site. And let's just move on a little bit because I don't want to, no, it's not letting me. All right, so we'll come back we'll come back to this and the importance of where the antibodies bind in a short while. But this just shows you that the antibodies bind to one particular region of the TSH receptor, which is where TSH binds, right? So that you can measure these antibodies by their disruption of TSH binding, or you can detect them. Right? This just shows you the weak association between the TSH receptor and Graves' disease. Very small differences in my view, giving significant associations, but nothing to be useful in the clinic by saying, let's look at a polymorphism in the TSH receptor and predicting Graves' disease. Right? I want to move to infection. So for 100 years or more, infection is thought to have been the cause of autoimmunity, and the data is really poor, right? Really poor. I don't have any residents to pick on. It's a shame. Let me ask you what people thought was the cause of Graves' disease before the discovery of TSH receptor antibodies. Anybody ever thought about this? Do we have anyone interested in thyroid here other than Dr. Winters? No, it's a tough question. That's why I like to use it. It's a great question. If you go back, I think the autoimmunity really started in the early 50s. So if you go back before the Second World War, what were people thinking of what Graves' disease was all about? They actually thought it was a pituitary disease due to too much TSH-like activity, right? Because they couldn't measure TSH. And as soon as they started to measure TSH, they found that there was no TSH in the serum of patients with Graves' disease. All right, so I'm getting diverted here. Let's deal with infection because I've got a complicated thing to try and a principle to give you. So this would be the second one after the linkage and association principle that I hope you remember. So this is RNA-seq. Every journal you open these days, if you actually go past the abstract, they're measuring RNA these days by RNA-seq. It's a very powerful tool. What it does is it measures all the expressed RNA in the tissue, all of the RNA. So since we have about 20,000 odd genes, and most tissues, 5,000 or 6,000 are active, you end up with a lot of information, right, on all the RNA that's active. And this is a slide that just shows you how you do it. You have to collect the RNA and fragment it, right, and convert it to cDNA, and then there are adapters that are available that allow you to PCR each of these fragments, which are then sequenced and recognized, right, according to a known database. And when we did that in patients with Graves' disease, we found 3 areas of activation. Obviously, there's going to be a lot of immune response activity in an autoimmune tissue. Right? And we found a lot of stress responses that we've actually gone on. I won't have time to go into that too much, to learn that even in Graves' disease, right, the cells are very stressed, and stressed cells are susceptible to cell death. They're also susceptible to thyroid growth, to cell growth, to stimulation, right? When you actually put TSH on a thyroid cell, the cell becomes stressed. So those are the 3 areas that we found in the RNA-seq data. This is from the publication, I think, yeah. It just makes the point, if you look at purple, that obviously within the thyroid gland of patients with Graves' disease, even after treatment with antithyroid drugs and coming to surgery, still a lot of immune reactivity in the gland. But one of the areas that took us by surprise, which is why I've included these slides, is this. And that there's a lot of evidence within the Graves' gland for viral involvement. Right? We don't know what the viruses are, but there's, as you see here, dendritic cell maturation, virus entry, genes are active, caveola endocytosis signaling, NF-kappa beta activation by viruses, etc., etc. This is what you can get by analyzing all the active genes in a tissue. And so that brings you to the conclusion that maybe viruses are very important in autoimmune thyroid disease. We just don't know what the viruses are. This is some data on hepatitis C that actually can be grown in thyroid tissue. You can find it in certain patients, and you can actually grow get the virus to reproduce. So the field is open for anyone here from ID to really get to the role of viruses in autoimmunity. It's still a very poorly understood area. Let's turn now to thyroid antibodies. So clinicians regularly measure in thyroid patients these three antibodies to thyroglobulin, which is the large glycoprotein on which thyroid hormone is synthesized, thyroid peroxidase, which takes part in thyroid hormone synthesis, and the TSH receptor that I just showed you a model of. Antibodies to all three are commercially available and regularly measured. So there is a famous study called the Wickham study from where I trained near Newcastle in the northeast of England. It's a town outside the city where they followed patients for 20 years. Right? They did a study, found patients with thyroid disease and followed them for 20 years. And it's been reproduced a couple of times in other areas. And the conclusion from that 20-year data was that individuals with thyroid antibodies and a borderline TSH progress to thyroid failure at a rate of about 5% per year. Right? So I'm sure you have some patients, at least I do, relatively neurotic patients who come to see you just because they've been found to have a thyroid antibody, right? And they want to know what to do about it, and you haven't a clue, but you can't say that, right? What you can say is that the presence of thyroid antibodies in certainly the female population is common. It's about 20% in young females, and that the progression, right, to thyroid failure is very much dependent on your TSH level. And if the TSH is borderline, you're going to progress at 5% per year. So in 10 years, you have a 50% chance of needing to take thyroid hormone. And that satisfies most such patients. And then they'll keep coming back year after year, many of them. And I just saw a dentist recently, a young lady who's been coming back for 10 years, and her TSH is now 11. And I said, okay, it's time to treat. The normal TSH is 2 or 3, and it's time to treat. So the presence of thyroid antibodies are risk factors for autoimmune thyroid disease. Now, the second point, actually the third principle I want to mention here is these are not pathogenic antibodies to TPO and thyroglobulin. Right? These antibodies are a reflection of thyroid damage. Right? So the body makes antibodies often to tissue that is damaged. And you know that because when you examine them, they are they are not oligoclonal, they are polyclonal. Right? So they have IgG1, IgG2, 3, 4, right? There's no evidence of a disease, right? These are just reactive, like a measles antibody. It doesn't cause disease, right? When you immunize against measles, those antibodies are not causing disease. Right? So these are reflections of Hashimoto's. So I really get annoyed when people say that thyroid antibodies cause Hashimoto's disease. It's a T-cell mediated disease, not antibody mediated. The same with anti-GAD in type 1 diabetes. Right? GAD antibodies don't cause diabetes. T-cells cause diabetes. Okay? In contrast, the TSH receptor is oligoclonal, usually only IgG1, and is a pathogenic antibody that causes disease. So these are the three types. This is the slide I was looking for earlier after the model. These are the three types of TSH receptor antibody that we find in patients with Graves' disease. They are unique human antibodies, not seen in the animal kingdom. No Graves' disease in any animal unless you immunize rodents. So the one on the left is called the best fit antibody. This is the antibody that fits very well on that leucine-rich repeat ectodomain that I showed you modeled. I call these Barney's antibodies. For those of you who know New York City, Barney's is a very expensive store, right? And they sell really good men's suits that I've never owned. But they look good and they fit perfectly, right? Like a made to measure antibody. So those are the thyroid stimulating antibodies. They sit in the leucine-rich repeat area and activate the receptor as good as, if not better than TSH. Then there's the Sears antibody, right? Sears is in the news, isn't it? Anyway, I call it the Sears antibody because that's the sort of suit I buy. Looks okay, fits really badly, right? So the Sears antibody sits on the TSH receptor and can't activate it, but it blocks TSH binding so that you can detect it, right, in the same way as the stimulator by inhibition of TSH binding, but it's not actually usually activating, or it's a weak agonist, a weak stimulation. We actually, I make my salary these days pretty much by looking at a third group, which is not clinically relevant necessarily, but these are antibodies that bind to the TSH receptor, but they don't interfere with TSH binding, right? So they can't give you a classical signal, but they bind to this hinge region, which is the part that links the important ectodomain to the cell membrane, right, and the transmembrane signaling domain. And we call these antibodies to the hinge region, or neutral antibodies, because they're neutral in the sense that they don't affect TSH binding, and they're not clinically available for testing. What's interesting about them is that they do, in fact, initiate a signal, but it's an aberrant signal, right? And it's a signal that stresses the cell more than the other antibodies, more than TSH, and that increase in reactive oxygen species leads to apoptosis. And if you look in a Graves gland, you will find areas of apoptosis that are probably secondary to these antibodies. So evidence that these TSH receptor antibodies are risk factors is pretty obvious. They're not present in normals or in any other disease that we know, unless you have autoimmune thyroid disease, and they're not available in animals either. Even monkeys have been looked at to see whether they develop Graves' disease. The common complaint or question at the end is, well, I've got a cat that's hypothyroid, right? Doesn't my cat have Graves' disease? And the answer is no. The cat has multinodular goiter that is toxic, usually, with a nodule making thyroid hormone. So cat hypothyroidism is not Graves' disease. Now, the story of Duncan Adams, talking a little too much this morning, but the story of Duncan Adams is really interesting. So he discovered the TSH receptor antibody while working in New Zealand in a place called Dunedin, at Otago, I think it's called Otago Medical School. And he published his results as an abstract in the local medical school journal. And that's still the primary reference for the discovery of these antibodies. And how he did it is quite interesting. Do I have a slide on that? Oh, no. So what at the time, right, which is the early 50s, the thought was that Graves' disease was due to too much TSH. And so the assays, the bioassays for thyroid stimulating hormone became available when radioiodine was released after the Second World War. People started to look at radioiodine uptake in animals and then humans. And he started to use a TSH bioassay, which essentially was looking at radioactive iodine moving into and out of the thyroid gland. And he had the idea that, well, let's take a look at the TSH levels in Graves' patients. And he did a bioassay on his Graves' patients and found that the time sequence was totally different. That when he put TSH in, he got stimulation in an hour or two. But when he put his patient's serum in, he got nothing in an hour or two until something slowly came up. And it went on for 2 days, the stimulation of the radioiodine uptake. Actually, he was using release. And he called it the long-acting thyroid stimulator, right? He thought, I think at that time, this must be some sort of aberrant TSH. But then they found that this activity was in the IgG fraction. And so, a long story, it came to be clear that this was a TSH receptor antibody. Now, he found it in every patient using this very simple guinea pig bioassay. And he found it in every patient because he chose patients with the Graves triad. The Graves triad is patients who have Graves' eye disease, Graves' thyroid disease, and Graves' skin disease. Right? They are the patients with the worst disease. And it turns out they, perhaps obviously, they have really high levels of TSH receptor antibodies. So if you have a patient like that, they're a great source of serum for really high levels of stimulating antibody. And having known that, those patients are very difficult to treat. They certainly don't respond well to antithyroid drugs. As soon as you stop them, it's back. And it turns out that the concentration of the TSH receptor antibody is a very useful predictor of recurrence of the disease after antithyroid drugs. And I always like to show this slide, which is one of my first papers in 1977 from the Lancet. You realize how long ago that is, Steve? So this was a study using antithyroid drugs, Graves' patients, and then stopping the drugs after 6 months and looking for recurrence. And it's an index. So actually the positive results, the positive antibodies are here. And you'll see that many of the patients who had positive antibodies recurred, right? And many of the patients who didn't recur. But there's a crossover. So the test is only helpful. Measuring receptor antibody at the end of antithyroid drug treatment is only helpful if it's positive. Right? If the test is positive, there's no point stopping the tablet. If it's negative, you're sort of 50-50. Now, everybody asks for the last 40 years, why did you only treat these patients for 6 months? Right? And the truth is that the fellowship ended. And I had to finish the study, right? Not an unusual story. But there have been other studies that have supported this. And I think I do that on every patient before stopping antithyroid drugs. And I don't stop if the test is positive. Now this hamster, what do you notice about this hamster? Yeah. Usually I'm accused of squeezing the hamster, right? But it's not, it's a thyrotoxic hamster. And this hamster was immunized against the TSH receptor, right? A wild type, normal sequence TSH receptor. And you can induce Graves' disease. So on the left is a normal thyroid from a hamster, and on the right is one thyrotoxic, right? Really active. And from one of these thyrotoxic hamsters, we were able to find we were able to find the first monoclonal antibody to the TSH receptor, which has been a very useful tool to probe the receptor and learn more about it. This just shows you that by taking what we call Mount Sinai 1, small amounts of the monoclonal, you can induce cyclic AMP stimulation in a cell with a TSH receptor, as opposed to other antibodies that either block or don't have any activity. And when you expose thyroid cells to one of these TSH receptors, this is now a neutral monoclonal antibody rather than a stimulator. Here is a neutral, which I mentioned, binds to that hinge region of the receptor and stresses the cells. You'll see that the caspase goes really quite high here. This is a control. And by exposing these cells to a neutral antibody, you actually start to become pro-apoptotic. And this shows you the reactive oxygen species stimulated by this cleavage region or hinge region antibody. And that makes the final point that you get apoptosis. So let's try and deal briefly with pregnancy, because time is rolling along here. I'm sure you're familiar with diagrams like this in the postpartum, where some 10% of women develop thyroid dysfunction, if you look, usually within the first 6 months or so. And you can get a whole variety of responses, either classic Graves' disease or classic Hashimoto's, or more likely, what people term postpartum thyroiditis, which is really postpartum Hashimoto's disease, with a time of thyroid cell death causing hyperthyroidism, followed by thyroid deficiency, and then a return to normal. And the reason for this is that during pregnancy, there is, of course, immune suppression. I'm going to take this off if it keeps doing this. During pregnancy, I think I'm just going to take it off. I have the feeling this is this. Let's see if that stops it. During pregnancy, where you've got, as I mentioned yesterday, essentially a transplant in the uterus, the immune system has to be controlled because the mother's maternal T cell become highly activated in the presence of this foreign antigen. And so during pregnancy, there's immune suppression. Once the placenta is rejected, right, then in the postpartum, you have a biological response to that suppression. And most biologic systems after suppression become overreactive. And that's why you see this transient increase in autoimmune disease, not just thyroid disease, in the postpartum. So pregnancy is a real risk for developing postpartum thyroid disease, whether you had disease before pregnancy and it was suppressed during pregnancy, or just are susceptible. New onset Graves' disease. In fact, it's amazing how many patients, if you ask, had a baby in the year before their onset of Graves' disease. 20, 30, 40% often of young women will give you that story, that they've got a baby at home. This is a slide that I showed yesterday just to make the point that during pregnancy, there has to be immune suppression. And the Treg cells, those regulatory cells, go up during pregnancy, shown here. There's the controls, and these are pregnant normal women. But if women have thyroid antibodies, they don't seem to make such a good regulatory T cell response. And we think that that's a significant reason, right, why certain pregnancies in women with thyroid antibodies are lost. Here's just showing you the immune response to pregnancy with suppression and the postpartum exaggeration shown with thyroid antibodies. And this is the increase in Tregs during pregnancy that you see in normal patients. Okay, so I had to rush through that a little bit. But I do want to make the point that this T regulatory response failure, right, does lead to miscarriages in patients with autoimmune thyroid disease. The miscarriage rate is about twice normal, right? Although that's not what we're talking about so much today, but something to be aware of. Let's go One of the last areas I wanted to mention was fetal microchimerism. Again, another principle more than thyroid oriented, and that is to realize that any woman who's had babies has many of them have in their circulation fetal cells from their babies that are retained. Right? The number that are retained is quite variable, but they can be there for years. I think the longest one in the press is about 22 years later. It shouldn't make you feel uncomfortable. Right? You should be quite happy that you've got some of your baby's cells, make you feel warm and cozy, I think. Yeah? Anyway, there is placental transfer, obviously, of fetal cells. There's also placental transfer of maternal cells into the fetus. They're much harder to find. But the theory has been, or the concern has been, as to whether the residual fetal cells, which remember are 50% foreign, can in some way activate autoimmunity. Right? I don't think the answer's there. Right? It's a very interesting approach, suggesting that during pregnancy, while the immune system is suppressed, as I've just stressed, right, that following delivery, because the fetal cells are retained, that the activated immune system will then start to recognize these fetal cells and develop a disease. And that's the theory just shown here, that after delivery, right, if the cells are in the thyroid gland, for example, they will become activated when there's loss of immune suppression. And so-called bystander cells, which are your inherited T cells that are sitting in the thyroid gland, will become activated. And if you go in and look at thyroid tissue, you actually find cells, male cells. In this case, it's easy to look for male cells in women. Right? Male cells are sitting in the Graves tissue, right, of women who've had male children. So they're there, but whether they're doing anything remains uncertain. Right? Now, I'm running out of time, but I do want to mention sex at the end, right? Because thyroid disease is essentially a female disease. I don't think the women's movement have quite got hold of this, but the ratio is something like 8-1, right? So it's a female disease, and the question is why, right? And it's not just female hormones, because this ratio continues in the post-menopausal period. There we go. So 1 in 8 from one paper I've chosen there. And one of the explanations has been X chromosome inactivation. So it's another principle to learn, or to understand. And that, of course, is that because women have 2 X's, you can't have them both working at the same time in the cell, right? So one is suppressed, right? One X is suppressed, and men don't have to do that. And this is called X inactivation. And it's somewhat random as to which X is suppressed, right? And so about 50-50, right? It can vary between tissues. It's really interesting. You read about it. Different tissues can be different X chromosomes that are active. So generally in the population, in what we call a non-skewed female, it's about 50-50, the X chromosome inactivation. And a skewed female is one in which there's more than 80% of one particular chromosome used, right, in the tissues. Right? And if you look in autoimmune disease, and including autoimmune thyroid disease, here's some of our own data some years ago now, that these are normal controls, and these are patients with Graves' and Hashimoto's disease. And this is the degree of skewing. In other words, what percentage of patients, or what number of patients show more than 80% skewing. And you'll see it's 9 versus 27%. And that's been reproduced in a variety of studies. And so this may very well be important as to which X chromosome you actually use. And that would suggest that the gene susceptibility for autoimmune thyroid disease should be on the X chromosome, right? There's not a lot of data to suggest that, but the FOXP3 gene is on the X chromosome, and that's a gene that's very important in regulating T cells. And so that might be the explanation as time goes on, as the data gets stronger. So, in summary, this just summarizes some of the things we've been talking about. It's a long story, and I haven't been able to hit everywhere. I've tried to stress certain principles for those of you who don't like thyroid. That there are predisposing genes that you inherit. We don't know a lot about them, and those that we do know are rather weak. That suggests, I think, that there's a lot of environmental triggers going on. We don't know whether this is smoke that seems to be associated with Graves' eye disease, or viruses that I tried to hint at. That would be different viruses in everybody because we all have different HLA. But something between the predisposition, right, the environmental triggers, sets off a pretty well understood series of pathologies that end up with Graves and Hashimoto's, or if you're lucky, just thyroid antibodies. So let me thank my guys in the lab, in particular Ralph Latif, who's my lab chief and an expert in the TSH receptor, and Syed Morshed, who's done the neutral antibody apoptosis story, and Risheng Ma, who I haven't been able to tour. His work is on thyroid stem cells. I haven't been able to deal with that at all. And support from NIH for more years than I want to think about. So I showed this slide yesterday. So anyone who saw it yesterday is not allowed to say anything. But you can identify the animal if you wish. But what is this? Anyone know? Anyone recognize what this is? The solenoid, what's it doing? On a camel. So this is a camel delivering immunizations to Bedouin children in the Negev desert. And they're using a solenoid to power the refrigerators on either side of the camel. And I like it because it tells me that there is always a way, right? That when you're trying to deal with a problem at home or at work, if you sit down and think and you get some wise advice, you can always find a solution. And I'm going to leave that with you to contemplate. Thank you very much.
Thank you so much for a provocative talk. I have one question to start things off. So, are there strong associations between autoimmune thyroid and other autoimmune?
Yes. In fact, I was telling Steve, my next talk is at the American Association of Rheumatology next week in Chicago. And so there are a lot of associations with other autoimmune diseases. They're weak associations. The strongest is with diabetes. Right? But yes, yeah. It's so that if you look at the patients with autoimmune thyroid disease, these associations are rare. But if you, for example, go to patients with myasthenia, you're going to find a lot of thyroid disease. You go to patients with MS, you're going to find a lot of thyroid disease, right? So, because one is common and one is rare. But you do see those. It was wrong. Right. Yeah. Yeah. Okay. Well, okay. So the thyroid is one of the most vascular glands around. It's almost like the kidney, which is why you can only do a hemithyroidectomy. You can't take a nodule out because the gland is so vascular, it just bleeds and bleeds and bleeds. So there's no problem with the lymphocytes getting into the thyroid gland. Right? As far as injury initiating disease, there is some evidence of trauma making disease worse. It comes from plastic surgery on pretibial myxedema. The plastic surgeons like to take that horrible stuff out, makes it much worse. In Graves' ophthalmopathy, if you do surgery, cosmetic surgery, too early, while the disease is active, it comes rip-roaring back. Right? So there is certainly evidence that local trauma can make autoimmunity worse. But as a cause of the disease, that would be your virus phenomenon, right? That the virus goes in there, sets off inflammation, and then the local autoimmune propensity takes over. So that's as much as I can tell you. Yes, very nice question, right? So people thought, well, the 21 gene must be involved because Hashimoto's is really quite common in Down's children, Down's adults now, right? They're living longer and longer and longer. But the association hasn't panned out in the genetic studies, right? So we don't really know why they're connected. It has to be something on the 21 chromosome, I think. But, in the GWAS studies, which are pretty useless, so it's all negative. And even coning down on some of those SNPs is not shown any association. But there are no huge studies. You've got to have a large number of patients for those sort of studies, and that's not been done with Down's kids. It's a good project. Good project for a fellow who's willing to go into, it was easier in the old days when all those children were in homes. Now they all tend to be still at home, so it's much harder to do now. But yeah, very interesting. There's a literature on it. Thank you very much.
Thank you for entertaining us and for all of our for all of our guest speakers. We send you home with a Louisville slugger.
Yes, thank you very much. Is this allowed on the air?
No, it's not. And in fact, generally, if you're checking out, you will see there are several pumps.
I see.
So we will send this to you.
We'll kind of mail it.
Your name, Medicine Grand Rounds, University. So thank you again.
Thank you. Very much. You see, it's a time I should. That's the stuff my boys take in my shirt. This will stop them taking.
That's exactly right. It's got your name on it.
Exactly. It's the same thing.
I actually, I was impressed with July.